We might be tempted to consider that food intake regulation is a step-by-step system. However, this is not true. The integration of internal signals, hormonal, neuronal, and metabolic, and external cues, behavioral, and cognitive, regulate the food intake cycle. In the next section, we will explore only some of the hormonal signals involved in the food cycle regulation. During fasting, the stomach is empty and relaxed, and this stimulates the vagus nerve to signal the brain and stimulate hunger. Also, during fasting, the empty stomach secretes a hormone called ghrelin, which reaches the brain and stimulates the sensation of hunger. At this point, we are starting to feel an uncomfortable emptiness in the abdomen and our hunger grows. While we eat, our stomach starts to stretch and ghrelin is no longer secreted. And the vagal nerve will send another signal to the brain, stimulating satiation. The digestive tract will begin, and the nutrients from the food, transported through the intestines by the peristaltic movements, will reach and stimulate the enteroendocrine cells, positioned in different segments of the intestines. Proteins and fats will stimulate the duodenum to secrete a hormone called cholecystokinin, which also reaches the brain, stimulating and prolonging satiation. Stimulated by fat and glucose, the descendant colon will secrete peptide YY and GLP-1, respectively. Both peptides will reach the brain, prolonging satiation and suppressing hunger. GLP-1 also reaches the pancreas, where it stimulates the release of insulin in a glucose-dependent manner. Let's zoom in into the pancreas and observe the beta cell. Besides the GLP-1 receptor expressed on the cellular membrane, the beta cell also has potassium and voltage-dependent calcium channels. In the absence of intracellular glucose, the membrane of the beta cell is polarized, as the potassium channels transport the potassium ions outside of the cell. So, the number of positive ions inside the cell will be less than the ions in the extracellular environment. The activation of the GLP-1 receptor will trigger the activation of the glucose transporter and the conversion of adenosine triphosphate into adenosine monophosphate, and increasing the intracellular calcium concentration. The activation of the glucose transporter will allow glucose to enter the beta cell. Glucose will be used for energy production, which will close the potassium channels, leading to the opening of the calcium channels, triggering the secretion and release of insulin into the bloodstream. Insulin will allow glucose to enter the cells and will stimulate the adipocytes to release leptin, which, together with the rest of the hormones, will induce and prolong satiety. During this period of time, eating behavior is inhibited. Satiety will end as food digestion continues and absorptive signals disappear. After the digestive process ends and the nutrients are absorbed, the stomach will start to contract again and the vagal nerve will no longer signal satiation. The cholecystokinin concentration will decrease, and the effect will start to fade. No longer stimulated, the colon will stop secreting PYY and GLP-1, and the effect of these hormones will also fade. As glucose is absorbed by the cells, the insulin concentration will also decrease, triggering the decrease of leptin as well. The sensation of satiety will start to fade, and the vagal nerve will stimulate hunger once more. Ghrelin will also be secreted by the empty stomach, prolonging hunger. The food intake cycle starts again. To recap, ghrelin is the hormone stimulating hunger and inhibiting satiety, hence, it has an orexigenic effect. In contrast, cholecystokinin, PYY, GLP-1 and leptin act by suppressing hunger and stimulating satiety, hence, they have a norexigenic effect. The absence of nutrients stimulates the release of ghrelin. Protein and fat trigger the secretion of cholecystokinin, fat stimulates the release of PYY, and carbohydrates stimulate the release of GLP-1 and leptin. A very important information to remember is that different nutrients stimulate the release of different hormones. This means that, for the regulation of the food intake cycle, what we eat is as relevant as how much we eat. As calories derived from different nutrients produce different hormonal responses, it means that not all calories are created equal, and what we eat influences our ability to control our hunger and cravings. In people with normal weight, hunger represents only a small fraction of the food intake cycle. While in people suffering from obesity, hunger represents the biggest part of the cycle. Knowing that the food intake cycle is a hormonal regulated process, we can arrive at the conclusion that people with normal weight have normal levels of the hormones involved in the regulation of the food intake cycle. So, people with normal weight can easily control their cravings and reach satiation. In contrast, people suffering from obesity have an abnormal food intake cycle dominated by hunger. Hence, they have abnormal hormonal levels. People with obesity have a decreased postprandial secretion of PYY and GLP-1, so people with obesity will not reach satiation after eating fats and carbohydrates as easily as people with normal weight. Obesity is characterized by impaired secretion of the PYY hormone. A recent study designed to compare the gastrointestinal hormone secretions between healthy individuals or controls and people with obesity showed that people with obesity have a significantly decreased secretion of PYY. While all classes of obesity had impaired postprandial PYY secretion, obesity class 2 and 3 did not experience any postprandial increase in PYY. Another study showed that GLP-1 secretion decreases with the increase of BMI. The inverse correlation with the BMI applies for both short-term secretion of GLP-1 from 0 to 30 minutes, shown on the left, as well as for the longer-term secretion of GLP-1 from 0 to 120 minutes, shown on the right, for both men with green and women with pink. Overall, obese and overweight individuals had up to 20% reduced GLP-1 response to oral glucose. GLP-1 secretion also decreases with increasing waist circumference. The same inverse correlation between GLP-1 secretion and waist circumference was applicable for men and women for both short-term secretion, shown in the left, as well as for long-term secretion, shown on the right. In this study, the GLP-1 response was assessed after the administration of a glucose tolerance test. Altogether, the findings indicate that obesity is characterized by both a decreased GLP-1 secretion and a decreased secretion of PYY. We will soon see that people with obesity also have an increased insulin secretion, which will overstimulate the adipocytes to secrete leptin, so people with obesity will also have an increased leptin secretion. The overexposure to insulin and leptin will produce the development of insulin resistance, which will trigger type 2 diabetes, and leptin resistance, which will impair the brain's ability to suppress hunger. Another interesting study showed that basal insulin secretion is more than 50% higher in people with obesity in comparison with lean individuals. Even more interesting was that the insulin secretion rates after glucose ingestion were also greater in the obese than the lean group, even though insulin sensitivity was not different between groups. Serum leptin concentrations are also correlated with the percentage of body fat. Another study showed that, in humans, serum leptin concentrations reflect the amount of adipose tissue in the body, as there seems to be a direct correlation between the body fat percentage and the serum leptin secretion. Contrary to what we might expect, obesity is characterized by an impaired secretion of ghrelin, the hunger-stimulating hormone. A recent study also showed that basal and postprandial secretion of ghrelin were lower in all obesity classes compared to controls. The results of the study proved the inverse relationship between BMI and both basal and postprandial plasma ghrelin. A very important finding of the study was that the ghrelin secretion seems to not decline postprandially in people with obesity class 3. All the hormonal signals reach the brain, which is the processor of the hormonal signals, which play a key role in energy homeostasis.